Okay, so this video is meant to be your little introduction to enzymes. And when we look at our standards and our proficiency scale, this video is just meant to kind of get you into some of the keywords like enzyme, substrate, active site, and product, and introduce you to the lock and key model of enzymatic activity. So, how enzymes work is still open for debate. Scientists know that enzymes help with chemical reactions, but they don't know exactly how they work. But the leading hypothesis and the leading theory is this lock and key model, where they have to fit together perfectly in order for the chemical reaction to occur. So we're going to focus more on, like this assignment here, more along the lines of scientist 1 than scientist 2 with induced fit. So, chemical reactions. Coming back to your chemistry, you learned about these different types of chemical reactions last year, and you learned about reactants and products. We are using the word products as well, and instead of using the word reactants, we're using substrates. So these two types of reactions, oxidation reduction or redox reactions, and dehydration synthesis or hydrolysis reactions, are versions of these that are a big part of biology. But the big idea here, within living things, we have all of these taking place, but enzymes make them possible. These things aren't going to happen within living things without the assistance of enzymes. So when we look at this, enzymes a protein that assists in the chemical reaction, both breaking things down, decomposition reactions, and also synthesis reactions, putting things back together. So like I said earlier, the substrate is the same thing as like the reactants. So here's our substrate, here's our substrate. The active site is the location where these enzyme and substrates fit together. So the active site is the location on the enzyme where the substrate and the enzyme bind, and then the chemical reaction can take place. And the idea here is that they're weakened and lowers the amount of energy for the chemical reaction to take place. And whatever we get at the end are our products. So again, we can take one substrate, break it into two products. We can take two substrates and bond them into one product. So chemical reactions can involve the breaking of bonds, or they can involve the forming of bonds. And all of these drawings are something that you're going to have to do in order to demonstrate this lock and key hypothesis. But again, with the lock and key hypothesis, this model demonstrates that they have to be the right shape in order for it to actually work, just like a lock has to be, or a key has to be the right shape to actually take care of the lock. So what we're going to look at now is this great website where their scientific models are actually animations. So this purple blob here represents the enzyme, and this string of green balls here represents a substrate. They fit on the active site, loosens up the bonds, so a chemical reaction can occur, and the chemical reaction is the breaking of the chemical bonds. Again, we said chemical reactions can also involve forming chemical bonds, and in this way, again, the purple blob here is the enzyme. Here are the substrates. The active site is the little circles, half moons built in here. And when they bind, we now have a product. Okay? So it doesn't just break things down, enzymes can also help build things up in synthesis reactions. Now, this whole lock and key hypothesis is important because if the active site is not the same shape as the substrate, no chemical reaction will occur. Now, what we're going to see with some of our experiments is if we heat this enzyme, the protein, too high, we can denature it or change the shape of the active sites. You change the shape of the protein, now the active site changes, and then the chemical reaction will no longer occur either, which that's the big idea why heat stroke is so dangerous, because if your body temperature gets way too high, you can start denaturing or breaking down the shape of the proteins and the shape of the active sites on your enzymes inside your body, and you can die from that. Other ways that things can happen is we can have inhibition. Inhibition is a way of stopping chemical reactions from occurring, and certain drugs work this way. If a chemical reaction is happening too often, or if we want to block a receptor site for a hormone, we can take some type of a medicine which will actually land on the active site and block the substrates from being able to land. And it inhibits the enzymes, it stops the enzyme from being able to work. Sometimes we want that to happen and we take medicine, but sometimes there are things like toxins and different poisons that also bind up active sites, which inhibit enzymes and then therefore stop chemical reactions, which can cause death. Now, not always do inhibitors land in the actual active site and competitively inhibit. They can also land on another site on the enzyme, and then it changes the shape of the active site, which then, if you change the shape of the active site, we can't have the substrate land on the active site, which then actually causes the chemical reaction to again not occur. This little model here is absolutely wonderful, and it's something we're going to get into in class. But hopefully this short video will get you into using the words enzyme and active site on that enzyme, and understanding the difference between a substrate and a product, and just in general how the whole lock and key hypothesis works. So hopefully, just by watching this short little video, you can already demonstrate a fairly clear understanding of the lock and key model of enzymatic activity by being able to draw and explain a lock and key model. So hopefully that was helpful.